A big thank you to our panel today, Dr. Gura, Dr. Mundy, Dr. Carroll, Dr. Kinberg, and Kin. Thank you so much, you guys, for joining us. I see all your faces. I think we're ready to go here. We do have probably about 15 minutes or so, a little less maybe than we anticipated. So we're going to get to as many questions as we can today. To the participants and attendees, if there is a question that you posed that wasn't answered, we will do our best if we can't address it during this live Q&A to make sure that we address that before the end of the conference. All right, so I've been pulling some great questions here. So first one, I'm gonna send to you, Dr. Gura. What would be the recommendation for a situation, so if we're talking about a mixed oil lipid emulsion, we heard you say 3 grams per kilo per day to prevent essential fatty acid deficiency. We have a patient who's roughly 49 kilos and only getting 55 grams a day. Is this something that they should address with their prescriber?
Well, I'm pretty much a pediatric-based person, and so my frame of reference is more pediatric focused. With the adults, they oftentimes have more reserves. They may take some enterals. Their requirements are different than the children. So this is when they definitely should talk to their prescriber. They may want to consider, checking an essential fatty acid profile just to see where they're going, and then determine if there's even a need to increase the dose. There may not be. Everybody's different. And so, I mean, it might be the European dosing that's been done in adults is lower than what we use in the US. So it may be totally appropriate for your situation. So again, have that conversation with your clinician.
Fantastic. While we're on the topic of lipids, we're going to stay with you, Dr. Gura. There were a couple of questions surrounding various allergies and use of different lipid emulsions. So fish allergies, shellfish, soy, or allergy versus intolerances. Do you have any kind of general recommendations about the use or caution with certain lipid emulsions?
Sure. First off, if you look at any lipid emulsion product, all the manufacturers say the first time you get exposed to a lipid emulsion, you should be monitored for the first hour of the infusion because you don't know if you're going to react. These are from oils. There shouldn't be any protein contaminants in the products. But if, let's say, you're on a soy and you're doing fine and they want to switch you and you have a history of fish allergy or shellfish allergy and you want to consider switching, you wouldn't want to do that at home. You'd want to do it in a monitored environment. Typically, you either come into the either an infusion center or the hospital or a clinic where they can actually do that monitored infusion to see if you are going to react. I have seen patients anaphylaxis soybean oil. So I always err on the side of caution. So I mean, again, you'd have to have find out what exactly that intolerance is. Oftentimes, if you've been tolerating propofol and egg-based vaccines, and you're telling everybody you have an egg allergy, you may not. You maybe had an egg intolerance when you were a child from orally ingested eggs, so you got a rash around your mouth. So, you have to evaluate each one of these on a per patient basis.
Thank you for that. We do have a couple other questions, and I might kick this out to Dr. Carroll or Dr. Mundy. Some questions more about lines themselves, and you both kind of covered, catheter related complications. Is there a recommended or preferred lock for a Hickman line? We have, a question in there. Somebody who was using heparin and switched to vancomycin. It helped with the lower the, infection rate, but they've had to use more cat flow. So is that a double-edged sword, when we switch like that?
Yeah, I think in terms of the locks, we've really been struggling in the US and are hoping some of the products from Europe will be available. For our group, when we had high rates of infections, always used ethanol locks. So that helped quite a bit. But unfortunately, I think the cost of ethanol locks has gone up considerably. So that's led to some issues there. So in that case, then we have to try and use antibiotic locks and trying to cover, what sort of antibiotic, what sort of bacteria may have led to the last infection, and doing our best to cover those. So it's a very difficult situation right now, I think, in the US in terms of preventing future infections.
And I think there's a problem. Even the ethanol isn't compatible with all the different catheter types. You can get some dissolving of the catheter, but I think it's all trade-off. If you switch to the antibiotic locks and the ethanol locks, the thrombosis rate is higher, but maybe at the price of buying some increased infection risk. I don't think there's a perfect solution. We use mostly heparin, and with frequent infections, we try the antibiotic locks. We tried some silver catheters for a while. They're expensive, and we didn't. In an ICU study, they weren't that much better. And they're
along those same lines, we did have a question as well on just, is there a better type of catheter that's, going to be better in preventing infection? Do you have a recommendation on catheter type replacement?
I think in general, so it all kind of depends on the center, is what I've seen from reviewing the literature. At our center, what we have done is that it really depends on how long you're going to have the catheter in place. And so whenever we're thinking beyond somewhere on the 4 to 6 week mark, we've gone with a single lumen Hickman catheter. And I bring up the lumen component as well, because that's also very important in terms of infection risk. The more lumens you have, obviously, the more chances you have of introducing bacteria and getting infected. So our group has kind of, if we're thinking short term, very short term, then PICC line is okay. But when we start to get beyond that 4 to 6 week mark, at least in our center, we've seen lower infection rates with sort of these tunneled catheters. And then it depends on what the need is, but sometimes patients also on long-term TPN may also have other needs like chemotherapy, and they may already have a pre-existing port. And so for the long-term patients, we've allowed those to kind of stay in. And there's other considerations like if they want to go swimming and things like that. But generally, I think you just have to look at your center's experience as to what success they're having with minimizing these infections.
Our next one is for Dr. Kinberg. Can you talk to us about interpretation of normal serum copper levels with low ceruloplasmin levels and what you might do with that?
Right, so that's why it's important to measure that free copper to be able to adjust. Because by itself might not be accurate. So you really want to look at those free copper levels to look for a true copper deficiency.
Yeah.
The patients, as I mentioned, orally can be very challenging.
And so still with you, Dr. Kinberg, and actually this could be a more of a panel discussion. Given some of the recent changes in the injectable multi-trace element products that we've seen in relation to, meeting recommendations made by Aspen, do you know of any future changes that might be made to the multivitamin products in terms of the level of vitamin D being provided in those?
I don't know of any offhand. I don't know if the rest of the panel has any knowledge on that.
I haven't heard of any new products, and I know they're very difficult to manufacture, so we'll see.
They're available in India.
No crystal balls.
Yeah, you'd be surprised what the rest of the world has that we don't.
Ken, there was a question for you that we didn't address earlier. I know we grabbed one of your questions. You had mentioned some resources that you had access to that sort of helped with getting genetic testing completed. Is there anything top of mind that you can share with our participants?
We were able to get our testing done through the University of Chicago in 2008. So they have a genetic testing lab, and if you call their lab manager, and if you have, so for us, I was able to, okay, so let's just, full disclosure here, I used to be a research scientist, and I've spent the bulk of my career as a regulatory affairs scientist. So I've spent a lot of time filing applications with the FDA. So when I
talked to you before, you're still a research scientist.
I'm just researching an N of 1 now, right? So when I called the lab at University of Chicago, I had the paper that Muller et al. Had published regarding the MID identification, which gene that was. And I was able to say, listen, can you run this protocol for us? Is there like a swab kit that we can send you, or is there some kind of way to get samples to you safely and in a controlled manner? And can you do this? And it took her, a few weeks to get back to us, but she was like, yep, we can do this. And so I know that at least the lab manager in 2008 at U Chicago, they're very open to that. So if you have a protocol and if you know what you're looking for, certainly the University of Chicago can do that for you. I think there were 3 or 4 other labs at the time that were also open to this kind of sort of commercial one-off testing. My guess is at this point, a lot more can do it. It's so common anymore. So I will find my contact, and I can post it to you at Oley if you want to share it back with the original questioner.
That would be fantastic. Thank you, Ken. Dr. Mundy, maybe this one might be for you. I mean, you talked a lot about the various, complications with HPN, and certainly there are so many. You covered the main ones. We do have a question in the Q&A related to the gallbladder. And so specifically, a patient with sludge and presence of a small stone, it's, in an HPN dependent type patient. Is this something that that should be monitored? Does the gallbladder need to be removed right away? What might be a kind of clinical course for this?
Yeah, absolutely. I mean, I think if you think about it, the gallbladder is kind of this bag that holds, bile, and then it's released as we eat to allow us to be able to digest food. In many cases with intestinal failure, patients are often not eating that much, or so you're missing some of that hormonal communication that releases that bile. So many patients are apt to have sludge develop. And then that sludge, as it accumulates, there could be a nidus and a stone can form. Now, obviously, it's not a guarantee, but sometimes these stones, as they get bigger and bigger and they're trying to pass, can go down the tract and cause a blockage there. And this can lead to pancreatitis and all sorts of, bad effects. So in terms of monitoring, yes, absolutely, it should be monitored. But then I think really the discussion should take place with, the team that's managing this individual to consider, do you use medications, such as ursodiol? Is it dire enough situation that you do have to remove the gallbladder? So I think that discussion needs to happen individually with the providers and the patient, so that they can go through all the options. But absolutely should be monitored, and in rare cases, this can lead to complications. So.
Great. think we might have time for one more. There's lots of catheter questions, such a hot topic. When would you guys recommend changing a line when it becomes occluded because of positional issues?
Yeah, that's so with positional issues, you mean where the line is located is causing a blockage?
I think that's what I'm interpreting from the question in the Q&A. So I would probably answer it that way. A collusion related to a positional issue. Yeah. Yeah.
So it's a tough call again. And I keep, almost kind of putting it back on the clinical team, but that's usually what we do. So whenever there's situations like this where, patients having difficulty flushing or the line doesn't work as well when they're in a certain position, we have our interventional radiologists take a look. Our mantra is always to preserve that line as long as possible. And I talk about the repairs, the catheter salvage for infections, so we do everything possible so that we're not constantly replacing, that line so that in the end, we don't lose access to that site. So that's the first thing we do, is that we just have interventional radiology evaluate the catheter, and they can recreate some of those positions and then see if the catheter could be adjusted. With a tunneled catheter, that's obviously harder, but they try and do that first before, we go ahead and remove it.
I was just going to add, sometimes I've had patients where they can flush the catheter, but they can't aspirate blood from it, and that's typically a fibrin sheath. And our IR guys think if that's a problem, that the fibrin sheath is kind of like a biofilm, and they like to change the catheter if that problem's encountered, because they usually find they're kind of fairly large looking when you see them sometimes come out. The sheath sits over the top of the catheter and the fluid will push through, but when you try to draw back blood, the sheath will occlude the catheter.
Just want to add one more. Yeah, just want to add one more thing on that. So when you do replace the catheter for something like a fibrin sheath, it's very important to have interventional radiology do it over a wire at the same site. That way you're preserving that vessel.